Type 1 diabetes is a condition where your body fails to produce a sufficient amount of insulin. Insulin is essential hormone that helps your body convert sugar from diet into energy. So in type 1 diabetes, the immune system mistakenly attacks and destroys the insulin producing cells in the pancreas. And this leads to an increased blood sugar levels. So in our study, we used experimental models to demonstrate that the gut microbiota and the immune system both are involved in the development of type 1 diabetes. So first, we showed that the transfer of microbiota from diabetes-resistant mice into diabetes-prone mice via a co-housing system reduced significantly the incidence of the disease. This microbiota transfer also modulated the recipient gut microbiota and their immune responses, shifting from a more harmful towards a balanced immune response. So this shows that gut microbiota can modulate the immune system. So in parallel, we also showed that the gut microbiome composition can vary according to the immune environment or the genetic backgrounds of the mice. So for instance, certain bacteria such as segmented filamentous bacteria were present in mice that lack B and T cells, whereas they were absent in immunocompetent animals. So this also shows that the immune system can modulate the gut microbiota. So to dig deeper in the pathogenicity of this disease, we showed that diabetes-prone mice exhibited a higher gut permeability, whereas those co-housed with diabetes-resistant mice showed an improved gut permeability. So this suggests that microbes or microbial products may translocate from the gut to the systemic system due to an impaired intestinal barrier. So altogether, our findings highlights the potential use of the gut microbiota to modulate the immune system and reduce the risk of type 1 diabetes.